We live in an era where we can edit human genes with CRISPR and print mRNA vaccines in a matter of days. But if you are bitten by a highly venomous snake today, your life depends entirely on a manufacturing process that relies on farm animals. The core recipe for the only proven cure, antivenom, was established by a French physician named Albert Calmette in 1894. The underlying method has barely changed since. Conservative estimates suggest that snake bites cause at least 81,000 deaths worldwide every year, with some figures climbing as high as 137,000. But the fatalities are only the surface of the crisis. For every death, roughly 3 more people, about 400,000 annually, suffer permanent, life-altering disabilities. This burden is carried almost entirely by the Global South. South Asia bears the heaviest load. India alone accounts for more than half the global total, losing an estimated 58,000 people to envenomation every year. Snakebite is one of the most devastating medical emergencies on Earth, yet the system designed to treat it is failing at every level. The financial reality of this drug is a study in extremes. In the United States, a single vial of the antivenom Crofab lists for over $3,100. Because a severe bite often requires a dozen vials or more, patients have walked away from hospitals with bills exceeding $140,000. But in sub-Saharan Africa, the problem isn't the price, it's the supply. Pharmaceutical companies have discontinued highly effective drugs, like Sanofi's Favafrique, because the market was no longer profitable enough to sustain production. When legitimate manufacturers pull out, the market fills with cheap, fake, or biologically inappropriate products. In some cases, antivenoms designed for Asian snakes are sold to African victims, providing a false sense of security as venom destroys their bodies. How can a drug whose main ingredients are simply snake venom and horse blood trigger such total market failure? To understand the scarcity and the ruinous price tag, we have to look at the unscalable 19th century manufacturing bottleneck still used to make it. Antivenom cannot be fully synthesized. It requires the raw, biological poison. Technicians must manually restrain deadly snakes and coax them to bite through a membrane to milk the venom, 1 drop at a time. The yields are minuscule. For smaller vipers, it can take over 125 snakes to produce a single gram of freeze-dried venom. That scarcity turns the powder into liquid gold, with prices running into the thousands of dollars per gram. This venom is then microdosed into farm animals, almost always horses, which act as living antibody factories. Horses are the industry standard because of their massive 40 to 50 liter blood volume. Instead, they mount a strong, sustained immune response to the venom and can serve for years across many immunization cycles. But an immune system cannot be rushed. It takes between 2 and 8 months of escalating injections to build a high enough concentration of antibodies to harvest. The global supply of this life-saving medicine hinges entirely on turning a living animal into a slow-moving biological refinery. Once the horse's antibody levels peak, technicians draw liters of blood from the jugular vein. The antibody-rich plasma is carefully separated, and the red blood cells are returned to the horse to speed its recovery. To make this horse protein safe for a human, technicians use enzymes to physically cut the antibody molecule. This process snips off the bottom tail, the part most likely to trigger a dangerous allergic reaction, leaving only the 2 binding arms that neutralize the venom. Because liquid medicine requires a strict cold chain, the purified antibodies are often freeze-dried into a stable powder that can survive transport to rural clinics. This process is incredibly region-specific. Because venom composition varies by species and geography, antivenom raised against Indian snakes is often useless against African species. This 12-month manufacturing cycle, combined with complex logistics and the risk of severe allergic reactions, is the reason the drug is so easily discontinued and so expensive to buy. After 130 years of stagnation, modern biotechnology is finally attempting to replace the horse. One major breakthrough involves broadly neutralizing human antibodies. By studying the blood of a self-immunizer who survived hundreds of bites over 18 years, researchers have isolated antibodies that can bind to toxins from many different species at once, potentially providing a blueprint for lab-grown antivenom. A second strategy uses tiny oral chemical inhibitors, like varisplatib. These molecules rush through the bloodstream and bind directly to specific venom toxins, potentially allowing a victim to take a pill in the field immediately after a bite to buy time. However, these modern solutions are currently limited to certain types of venom, such as those from cobras and mambas. They do not yet cover the tissue-destroying venoms of vipers, and they remain years away from large-scale clinical use. While the biotech future is promising, it isn't here yet. For the foreseeable future, the survival of hundreds of thousands of victims will rely on the quiet Victorian alliance of a little bit of poison and a patient horse.